Well, hello, good afternoon, everybody. We're thrilled to be here and so thrilled to have Dr. Collins with us. I know he needs no introduction, but I will share his intro with everybody. So Dr. Francis Collins is the 16th director of the NIH. He was appointed by President Barack Obama and was sworn in on August 17, 2009. In June of this year, President Donald Trump announced his selection of Dr. Collins to continue to serve as the NIH director. And in this role, Dr. Collins oversees the work of the largest supporter of biomedical research in the world, spanning the spectrum from basic to clinical research. Dr. Collins is a physician geneticist noted for his landmark discoveries of disease genes and his leadership of the International Human Genome Project, which culminated in April 2003 with the completion of a finished sequence of the Human DNA Instruction Book. He served as director of the National Human Genome Research Institute at NIH from 1993 to 2008. Before coming to NIH, Dr. Collins was a Howard Hughes Medical Institute investigator at the University of Michigan. He's an elected member of the National Academy of Medicine and the National Academy of Sciences, was awarded the Presidential Medal of Freedom in November 2007, and received the National Medal of Science in 2009. We're so honored to have you speak to us today, Dr. Collins, to share your thoughts and experiences in science, especially around team science. So let's get started with hearing about your early career experience. What made you choose human genetics?
Well, that's a tangled tale, but I'll try to tell it quickly. But first, I just want to say how wonderful it is to be able to be part of this program today. I'm a big fan of the Birch program, which has done a lot to support junior faculty and trainees who are interested in women's health as an area of research, and has resulted in creating quite a wonderful cohort of remarkable investigators that are really pushing back the frontiers. I also do have to give a shout out to Igo Afodokun, my friend. We met 24 years ago when he was a resident in a hospital in Nigeria, for which I was serving as a volunteer, temporary 2-week rotation internal medicine attending. And it was pretty clear Igo was somebody of exceptional talent. And for various personal and professional reasons, things happened that resulted in, well, the rest is history. There he is now as a remarkably effective and productive faculty member at Emory. So Igo, hats off to you.
Thank you. Thanks.
So what got me into human genetics? Well, I didn't start out that way, that's for sure. And maybe there's a lesson here in terms of anybody who's trying to map out their career. If you think you're going to know where you're going for more than a year or two from where you are, probably some surprise will come along, because that seems to be the nature of things. I didn't have any interest in life science at all. As an undergraduate, I went off and did a PhD in chemical physics, avoiding anything to do with life sciences because they seemed terribly messy. But then I discovered something was happening in life sciences with recombinant DNA, and that actually life made sense. And there were aspects of this that were incredibly satisfying from a mathematical perspective, because I really liked math. As a result of that, I changed my direction, decided to walk away from the very academic pathway in academic physical chemistry, and went to medical school. But I wasn't quite sure how to put that all together until on a very small series of lectures about human genetics as a first-year medical student, I realized that was my sweet spot. It was about life, it was about people, but it was also about something that I could really get excited about, which were the principles of how life works with nucleic acids, DNA, RNA, and protein. And I was hooked. And of course, it took a long time to turn that into something you might actually call a life plan, but over the space of time, through medical school, through residency in internal medicine, I was even a chief resident, then a fellowship in medical genetics, ultimately a faculty position at Michigan, and then getting the call to come to NIH and lead this amazing thing called the Human Genome Project.
So you highlighted some variations along your early career path, but are there any other challenges that you would like to share with us as you started that career?
Well, some of it was just trying to figure out how to learn something completely new when you were abandoning the things you were good at and jumping into a space you knew nothing about. There's something about career training that seems to go through those series of transitions where you get into something, you get really good at it, and then just when you think you have some self-confidence, you pick another field and discover you're really lost. And I had those repeated experiences, and I bet anybody who's listening to this, who's had that same opportunity to change directions a little bit, knows what I'm talking about. But it's also exhilarating to spread your horizons and take in information that might have been way outside of your area of interest, and now it becomes really fascinating.
So on that note, did you anticipate the leadership roles that you've held so far? And if so, how did you prepare for them?
Well, I certainly did not. I never imagined myself as being in charge of anything much bigger than, say, a small research laboratory. And in fact, I wasn't in charge of much of anything larger than that until that call came in 1993 to come and lead the Human Genome Project at NIH. I was quite uncertain that this was the right thing for me to do. I didn't think I necessarily had the skills to be a manager on that scale. I wasn't sure the Genome Project was actually going to work. In fact, most people thought it was probably going to be a big, messy failure because the technology wasn't up to the task. And I wasn't sure I wanted to step away from what had been a very satisfying role, running a research laboratory working on human genetic diseases like cystic fibrosis and neurofibromatosis and Huntington's disease, and taking on something that was going to be a much more complicated effort involving potentially hundreds of scientists in multiple countries in the world. Was that really where I was going to find my own skills fit the circumstance? I talked to my mother about this, because one should speak to one's mother when you're contemplating such a career change. And she didn't help me much, because she said, you know, I'm proud of all 4 of my sons, and I always think that they should do the thing that is going to give them the greatest chance to make a contribution. But one thing I've always said is that I hope none of my 4 boys would ever become a federal employee. Which, of course, was what I was being asked to do, because if you were going to run the Genome Project, you had to come and run a center at NIH. It was one of the few times that I disobeyed my mother, and I'm glad in this case. One time in her whole life, she was actually mistaken, because this was, it turned out, the most amazing experience that I could have imagined. But certainly not something that I planned, lobbied for, reached out for. It just sort of happened. There was this opportunity. And I actually said no the first time it was offered. And I thought to myself, there's only going to be one Human Genome Project. This is only going to happen once. Okay, so it might be a big flop, but it might actually succeed. And would you want to miss the chance to kind of stand at the helm and lead this forward? Okay, I'm in. I said yes.
So you've talked a little bit about the Human Genome Project, and it's been one of the largest team science endeavors of our time. So you talked about some of the early challenges with it, but what do you think contributed to your being chosen to lead the group?
You know, it got off to a start in 1990. As I mentioned, there was a lot of controversy about whether this was a good idea or not, and more than half of the scientific community was opposed. They thought this was technologically premature, that we wouldn't figure out how to sequence 3 billion letters of DNA. Gosh, at that point, you could almost get a PhD for sequencing 1,000 letters of DNA. So the scale was just breathtaking to imagine you could achieve that. There were big concerns that it was going to have a big cost overrun, and it would take money away from other much more meritorious investigator and driven research. It was going to become just a sink for all kinds of money going down the drain. There was a particularly irritating concern that only mediocre scientists would want to work on it, because it was just going to be so mind-numbingly boring reading out all those letters of the DNA code. They even proposed that it should be assigned to prisoners, and they would get less time in jail if they would sequence a few thousand base pairs. So it was not an easy time. Now, the project got underway largely because of the strength of leadership of a small group of very visible scientists, most notably Jim Watson himself of Watson and Crick, who agreed to be the first leader. And he had a great ability to convince skeptics in the scientific community and to convince the Congress that this was something that we should do, and therefore they should appropriate some money. But he didn't stay very long. He ended up in a tangle with the current NIH director at that time, Bernadine Healy. Jim Watson was never one to be shy with his opinions, and after he referred to his boss, the NIH director, as a lunatic in front of an open microphone several times, that kind of didn't look like it was going to work anymore, and suddenly Jim was gone. So there was this big debate. Oh gosh, what happens now? This project is barely getting off the ground. It has all these risks. Who's going to be able to do this? And somebody felt like it would be good if it was a physician scientist who could talk about the medical implications, because maybe that would sustain the interest. And there weren't that many physician scientists involved in the Genome Project. There weren't that many people involved at that point. So I guess that's why my phone rang.
It's so interesting to hear the background story. Of course, most of us don't know all the ins and outs of that. So thank you for sharing that part of it. I can imagine, as being the new leader in this, that putting together a team was really, really important. So how did you go about putting together the team to take on this new project?
Well, I wanted to reach out and find the best and brightest of this generation of biologists and geneticists to try to convince them what I had become convinced of, that this really was the flagship project of a generation. This was something that, if we succeeded, would bring us entirely into new territory of an understanding of our own biology. And everything would be different after that. And if you had a chance to be part of something, why wouldn't you? So a big part of it was that kind of outreach. And it was a pretty compelling story to be able to tell. And that meant asking people who are planning to do something else, that this would become their passion. And over the course of a few years, that network of leaders across the country and across the world really came together. And I had wonderful help in that regard. And as far as my own staff, I tried to identify the smartest people I could find, and I tried to empower them to have the kind of independent thinking that we needed in every step. And I particularly told them, the one thing you must never do is to hide information from me that you think will upset me, because I've got to know. If something isn't working, the sooner I know about it, the better. And by the way, also, if you think I'm about to make a mistake, tell me. I want everybody around me to have the freedom, the permission, the encouragement to tell the director that he's about to do something really stupid, because otherwise, why do I need you all here? And they took that literally, and they frequently did tell me I was about to do something stupid, and most of the time they were right. So it was a wonderfully interactive, democratic kind of system. And then we had to figure out how to manage this, because ultimately there were 2,400 scientists working on the Genome Project in 6 countries, in 20 laboratories. And they all had to come to an agreement about what the goals were and put their shoulders to the same wheel and have the same standards about accuracy and assembly. That was a challenge. So we had to get agreement to the standards. And then we had to figure out how to do the management. And a lot of that, it was done by phone calls, frankly, because that was the way to get everybody in real time. I still, at 11 o'clock on Friday morning, feel like, oh my gosh, I should be ready for the Genome conference call, because every Friday at 11 o'clock, we would have people all over the world signed on. And we tried to make it both a hard-nosed management discussion about how many base pairs have you sequenced and what's your evidence that your accuracy is right, but also turn it into a lab meeting where people could talk about some new cool technology that they had just started to put into their pipeline. And it got to be quite a community. Not to say there weren't frictions. There were times where I had to kind of take people to the woodshed who were behaving badly, but most of the time we were family, and we were devoted to the idea that this was a historic opportunity that would probably never come along again, and we were all part of it.
That's amazing. So that is team science on a scale probably none of us could imagine.
Yeah.
That's huge. So on that message of teamwork, that was probably one of the biggest examples of a major research project with such a huge team. But how did that shape your view of teamwork, and why do you think teamwork is so important to science rather than the old model maybe of solo investigators?
Well, I think teams are the way in which you can accomplish some very complicated efforts, but I don't want to overstate this. I still think most of the discoveries that will be ground shaking in the next decade will come from individual investigators who know how to utilize this remarkable availability of technologies and free databases to make discoveries. So if people are listening thinking, oh, well, you know, us folks who aren't part of a big team, we're not important anymore. Not true. The real initiative, the real intellectual leaps come from those individuals, but we want to empower them. I think that's what teams do. the way I think of this at NIH is there are certain kinds of projects which could actually provide incredible power to lots of investigators all over the world, but they're not the kind of thing that are just going to happen because somebody sends us a grant application. They're going to require bringing together a larger group with different disciplinary skills. Look what we're doing right now with the BRAIN Initiative, which, talk about audacious. I think we want to understand how those 86 billion neurons between your ears do what they do. That's the most complicated structure in the known universe. Certainly makes the Human Genome Project seem like child's play by comparison. And yet we're determined to try to figure that out. And if we didn't do anything to organize team efforts, ultimately, over the course of many months or years, we might actually make some progress. But if we could figure out a way to inspire more of that by setting up a program with managers and teams and opportunities for people to share information and have everything open access, it'll go faster. And that's kind of what we're doing in that space. And I could cite several other examples like that. But again, that's a small proportion of NIH's research investment. It tends to get some visibility because these are big, complicated projects that, if they succeed, are going to have a lot of implications. But most of the work that's going on right now is not of that sort. Now, that being said, individual investigators, I think now, often do find themselves working in more of a team orientation than they did 20 years ago. My own lab, which is like 200 yards from here, we work on diabetes. We work on this premature aging condition called progeria. In both of those projects, we have collaborations, both here at NIH and with investigators in other institutions. And we have very much then a team spirit about, okay, you do this part, I'll do that part. We'll both get there faster because we can share the experiences that each is able to bring to the table. And that's good. And that basically means everybody has the chance to get involved in science that they couldn't have done by themselves. Mm-hmm. We all have to be thoughtful about that. This is one of the things that many of us involved in teams have tried to pay attention to, that being in a team of this sort should not diminish your own personal satisfaction or your personal recognition. And the people leading teams need to always be thinking about who else is involved, especially the junior people, and making sure they get the credit and the visibility for being part of such an effort. Looking back on the Genome Project, where we worried about that a lot, I think it turned out really well. Of the junior scientists that were part of the Genome Project, I can sort of point to what happened to them. They've all flourished and built upon having been at the center of that activity to fashion subsequent career paths that have been enormously successful.
So you mentioned junior scientists on a large team, and so what would be your suggestion for how to get junior scientists more visible and more active in these large team science projects?
Well, again, it's up to the sort of organizational structure of the team, and particularly the senior people, to pay close attention to that. And so when there is a group that needs to be spun off to work on a subset, make sure that the junior people are given a chance to lead those smaller groups, even if they're not at the point of being able to lead the whole enterprise. Thinking ahead about publications, because that's still the currency that determines a lot of recognition in academia. Which publications could be sequenced in which order, and who could actually be sure they had the appropriate recognition in the author list for a publication where they played a significant role. I gotta say, the use of being able to have joint first author positions, use of asterisks, has helped a lot in that regard, because there used to be, before we did a lot of that, some real harm that got done when you had a team and there were a lot of people who'd made significant contributions and just one name was going to be considered as having driven the whole enterprise by being first. Now, I'm happy to see papers where there are 2, 3, even 4 or 5 asterisk first authors of people who had equal contributions. That's sort of how the science gets done now.
Mm-hmm. So you alluded to this a little bit earlier when you were talking about especially the large team with the Human Genome Project, but how did you manage challenges among the team, on your current teams and on the big team? So bad behavior or some of those things that you sort of mentioned.
Well, people are people, and people don't always have the ideal behavior patterns, especially when we're talking about people who are competitive, as many scientists are. And again, I think it's kind of up to the team leaders to watch for circumstances that are shifting away from the completely healthy and try to head them off. And I certainly did find myself at times feeling a bit like a counselor to group therapy as opposed to a scientific project manager. And particularly in the Genome Project, let's just be clear, there were some egos involved in some of the major leaders that were world class in terms of what they expected, and particularly their being used to being in charge and not having anybody else tell them exactly how to run their operation. And we had to get past that, or we were not going to have a Genome Project. We were going to have a whole lot of fiefdoms that didn't get along. And that took a lot of personal time. You know, what really is the way to get past that is if everybody has bought into the mission, the goal, and they can see how critical it is for that goal to be achieved, that everybody on the team has to succeed, not just them, then you can often get past a period of some friction and get everybody reoriented, working towards that same vision.
And how do you know when to just make a cut? When the relationship is no longer working, how do you make that decision?
Oh yeah, I hate that part, but it does have to happen. With the Genome Project, you know, we started off with some 20 laboratories involved in this effort. And as the scaling up started to happen, not everybody could do it. And we had to drop a few off. And those were some of the most painful things that I had to do, is basically call the head of that team, who's standing in then for quite a lot of people, and say, you know, thanks for a great run. We will certainly use all the data you've generated. But at this point, I think it would be better for your team to find some other kind of focus, because we've kind of run past the point where you are, and I don't think you can catch up. I still remember those calls. It was the right thing to do. And I think anybody who's been involved in large organizational structures has to have had those experiences, but they're not your favorite part. At least they're not my favorite part. Maybe there are people who enjoy that, but I don't.
And then on the more positive side, how do you, what are your strategies for motivating a group to create synergy and really work together well as a team?
I think a lot of it is to figure out a way to shape an enterprise that captures the imagination and the passion of all the participants, so that the goal becomes sufficiently compelling that you can set aside some of the other complaints that you might have had about the sort of daily activities involved. It was not so hard to do that with the Genome Project. After all, it was, as soon as you got your mind around this, about how historic it was, that was a vision that anybody involved in the project had to agree to and couldn't really question. Other teams doing other projects don't necessarily always have something that is as universally compelling as that. But before you ever even start, you better find out, is your vision good enough that it's going to sustain the whole group over those rough spots? And if not, maybe you better think hard about whether there's a way to redefine it or change some of the parameters so that everybody signs on saying, yes, this is the thing I really want to do. I'm prepared to sacrifice maybe some of my own personal desires about being in charge for the larger goal of getting this science done and making a discovery or a new data set that everybody's going to want to use a real possibility.
Great. So you talked a lot about senior leadership and team leaders really recognizing junior investigators, but what would be your advice for junior scientists themselves to be successful at team science? Like, what are some strategies we can learn or implement to be a better team scientist?
Well, first of all, is to figure out what kind of a team effort is in your sweet spot, because not everything you get invited to take part in as far as a team project is necessarily going to fit with where your own skills and dreams land. So it's always possible that when you get invited to one of these, that the answer should be no. This is like, thank you very much. I'm flattered that you wanted me to take part, but actually my dreams are in a different direction. So be selective. I think sometimes, particularly junior investigators, feel like I've got to get my whole portfolio of activities spread in many different directions because I don't know which ones are going to pan out. But then pretty soon you're so diffuse that you're not making much headway in any direction. So pick carefully. And I guess I would say, if you're going to be involved in a team or even your own individual project, pick a question that really matters. if it's a team that's doing something, which if it succeeds, is really going to shed new light on our understanding of life and how disease occurs, that's pretty exciting. If it's a team that's going to do the next obvious incremental step, even though they may generate some obvious data that's publishable, is that really where you want to spend your time? We don't get that many years here to be a productive scientist. You may as well spend your time on something that's a question that really counts. Even if it's a little riskier, it's in the long run probably a better way to make your contribution. So be selective. And then once you've selected, be both incredibly collegial and congenial and friendly and flexible, but also make it very clear what your area of expertise is. Make sure that then as the team comes together, what is your part of it and what is everybody else doing? I've certainly seen teams where that was not well defined. And a few months along, it was discovered that people were actually doing the same thing without realizing it. And that makes everybody unhappy because time gets wasted and there's questions about who gets credit. So make sure the team really has its roles well defined. And then as far as who the other members of the team are, seek out teams where the other people are different than you in their expertise, in their sort of their philosophy of life, their perspective. We know that when you look scientifically at productivity, it's the teams that have diversity of all sorts that are the most productive. And if you have a team where everybody's kind of like everybody else, maybe you didn't need a team at all. You need that difference in perspectives. Look for those. So yeah, the right people, the right definition of the project, and a project that is exciting, groundbreaking, something that you will wake up in the morning going, I can't wait to start working on that one again today because we're going to learn something there that's going to change everything. That's what you want to do.
So that's great advice. There is a question on our agenda about carving out that independent project of a bigger team. And I think you alluded to it a little bit. And it sounds like communication is a real key factor, finding out what other people are doing.
Yeah.
And you talked about some of the challenges with that where there may be overlapping. Would you have any other advice for how to really stake your claim or really find your niche in a big team like that?
Yeah, again, the definition of exactly who's doing what and what you are doing, even to the point of talking somewhat into the future about how publications are going to be handled and who will be able to give the big presentation at the scientific meeting of your professional colleagues, so that it's not just sort of left unattended to. Many of these teams, I think, even have an initial working group that gets set up right away about publications and presentations, how that's going to be handled. If you have such a working group, it's good for junior people to be on it so that they have a chance for their views to be heard. And I guess also, it makes such a difference if you develop that kind of confidence in your team members, that they know that you're somebody that can be counted on, that you're not all looking out for yourself, and you're not going to be prickly and take offense when somebody says something that doesn't quite mesh with your own view, and that you're willing to take on additional work on behalf of the team if it makes sense for you to be asked to do so. You become the indispensable member of the team where everybody goes, I'm so glad I'm part of this because Mary is the person who's always got the sort of right attitude and keeps us on track. That makes a big difference. And oftentimes it's the junior people on the team that set that standard.
And so what would your advice to us be if we're trying to do all that and we're trying to be the best team member and maybe it's not the best fit? How do you navigate that?
Got to think about that too. And again, I've been in that experience of being on a team that seemed like it was going to be a really good thing. And then ultimately over the course of time, turned out that not everybody had really agreed to the stated goals. And there was not open sharing of information. There were sort of subgroups that were keeping information to themselves and the rest of the group not hearing about it. And pretty soon everybody stopped trusting everybody and like, oh boy. So you have to have an exit strategy in a circumstance of that sort. And that can be very painful, but it also is something, if you really reach the point where clearly this is not living up to the expectations you had, and you're feeling like you're putting time into something that's not going to turn out well, then it's time to call the question. Go to the most senior sort of leadership of the team, lay out your case, and ask for a pathway that would get you out of that circumstance, but still preserve what you've already contributed in terms of recognition, publications, and so on. Hopefully that won't happen very often, but realistically, it can.
Right. Thank you. Thank you for sharing that. Brittany, should I open it up to the panel or the audience to see if they have any questions about challenges with team science?
Yeah, I think it's a good reminder that to please go ahead and ask some questions.
I see we have 48 people watching. Of course, that's what my screen says. So bring it on, y'all.
Yeah, we'd love to hear questions from the participants. So feel free to send those in. As we potentially wait for that, I'm going to ask you a big question. So what was your greatest leadership challenge, either while you were working on the Human Genome Project, perhaps then, and also maybe now?
There have been a lot, I can tell you. I guess, I'm going to tell you a story which is probably the most painful one of my professional career, but I think it's one that I learned from, and maybe by walking you all through it, you will not make the mistakes that I made at that time. This was just about 20 years ago. I had moved my laboratory from Michigan to NIH and brought with me a small group of researchers, postdocs, and graduate students that had been working with me in Ann Arbor and were not quite finished with their projects and came here to continue the work. We were working on a bunch of different projects. One of them was on a particular type of leukemia, the so-called M4 type of acute myelogenous leukemia, where we had discovered what was the molecular basis of the common cytogenetic abnormality, which was an inversion of chromosome 16. And it was a pretty exciting finding because it basically flipped around part of chromosome 16 and brought together two genes that were supposed to be on different arms of that chromosome, causing them to make a fusion protein that was an oncoprotein and transformed those normal white cells into leukemia. And a lot of excitement, several people in my lab working on this, including a MD PhD student who was one of the most talented people I'd ever seen in my laboratory in terms of his vision, his ability to think through complex problems and design interesting experiments. And he was well along in his training part when we had sent a paper in for review. And the editor of the journal wrote back a personal note saying, I think you ought to look carefully at one of the figures, because there is evidence, once you know what to look for, that it's been manipulated. I was horrified. And when I looked at that figure, I could immediately see what was being talked about, which had escaped my ability to pick up on it, because, of course, I had assumed that everything was fine. That raised my concern. I went to the student. He said it was just a careless mistake in putting the figure together. It was done late at night, and he had made a mistake, and he could correct it. But there was something about what he said that didn't quite sound right, and caused me then to start looking at other things that he had done, including other published papers. And over the course of a week or two, it became very clear that this was a student who had massively fabricated a large amount of the data that he had produced as a student. Even though he was working closely with a postdoctoral fellow, no one suspected this. I didn't, neither did the postdoc, but there it was. So what went wrong? I'm not sure I completely could tell you why we were not sensitized to this, why we didn't discover it sooner, or even why this extremely talented student would take this terrible risk of making up data. And when you look at the experiments that we published, in almost every instance, the conclusion was right, but he was too impatient to actually do the work. So he instead produced the data to go on to the next experiment, just to speed the whole thing up. A horrifying situation. If there's anything that science has to stand for, it's that the data we produce is trustworthy, it's true. And here was the most dramatic aberration to that principle that you could imagine. So what to do? Basically, did the appropriate things that you have to do now when fabrication is discovered, convene a committee, have them look at all the data. Ultimately, the student was denied both his PhD and his MD. And I decided the only thing that could come out of this was to be extremely public about it. And so I began talking about it to anybody who invited me to a meeting. Instead of talking about my research, I talked about this experience and said, you all should learn from this. None of us are probably immune to this, but I probably made mistakes by being too trustworthy and maybe not asking to look at lab notebooks every day. The New York Times wrote up a big story on this, which was profoundly embarrassing, but it was still the right thing to come clean about. Mm. So talk about a professional challenge. When you're a scientist and you find that you've provided over the worst thing that you can imagine in your own laboratory, namely people fabricating data, that's a challenge. I still think of that. I still look at every figure that comes out of my lab asking, is there any way that could be manipulated? Which is totally unfair now to the people who work there who are entirely trustworthy. But that kind of sensitivity, it's probably got to be part of our professional view. We've got to be committed to the truth, no matter how hard it is to think about the fact that it might not always be there.
Wow, what a tremendous dilemma. Thank you for sharing that with us. Sort of on those lines, we do have a question from a participant out there. Bradley Richmond wrote in and asked, so in light of an experience like that, how do you keep other people motivated when things aren't going well? So you gave a dramatic example of a pretty ethical issue on research, but for the other members of the team or in another situation, how do you keep the team motivated, and do you tailor it to an individual who needs that?
Everybody's different, sure. In my own group now, about 10 people, I know I have some trainees who are absolutely going to be pushing every day. They come in excited. They work really hard. They crank out interesting observations. They occasionally stumble, but they pick themselves up quickly. And I know there are others for whom this is not such an easy pathway and may get discouraged. And oftentimes, we all have personal issues too. I think as a lab director, while you don't want to be poking into people's private issues if they want to keep them private, it also is sort of like your family. And so you have to think about, okay, who just had a parent who got a terrible diagnosis of some sort that's really on their mind today, and maybe that's why they're not quite paying attention to the details of that experimental protocol, and who is actually struggling right now with a career decision about whether they're on the right path or whether something different is coming along that might totally upset their apple cart in terms of what their life vision had been. Staying in touch with people about those issues. I try to meet with all my trainees one-on-one on a regular basis, and not just ask them to show me their data, but also to say, so how's it going? And find out if there are other things that are getting in the way of their being able to feel completely successful in the work that they're doing in the lab. That's just, I think, a good way to deal with real people who are working really hard to try to make interesting findings happen. Because science, as it has been said, science is hard. It's not like you just walk in and everything works. In my lab, I would guess on a good day, 5% of our experiments work, because most of the time they don't. And anybody who tells me their experiments are working most of the time, I'm going to say they're not working in a very interesting area, because things that are that reproducible and easy, generally, well, that's just crank turning. That's not really pushing back the frontiers.
Those are really good points. And I know in my own experience working on a team, the challenge with different responsibilities of different team members and getting everybody to buy in and still maintaining productivity. Mm-hmm. How would you advise junior scientists who are dependent on other members of the team to move forward? How to navigate that and stay productive? And
yeah, that can be a challenge for sure, especially if the team is not sort of equally carrying the load of the effort. And again, as a junior scientist, you may not be in the best position to try to change behaviors if somebody is not holding up their end and everybody else is therefore held back by that. That's really where the senior leadership of the team has to step in and help you out, which means you need to have the relationship and the confidence of the senior leaders, so that when something like this is happening, you can raise that without appearing to be a whining person. You're basically saying, look, this project, in order to succeed, has got to have all hands on deck, and there's not everybody quite buying into that, so help. And I think a senior leader who hears that will take it with great seriousness. Obviously, you may be able to encourage your colleagues without having to take it to that level of raising the concern to the senior leadership, simply by discussing with whichever part of the team you're concerned about is not holding up their end, and find out why that is. Mm-hmm. Maybe actually they're stuck on some sort of obstacle of a technical sort or a personal sort that nobody knows about. And if they did know about, the group could come up with a solution. Maybe you can sort of get this out in the open, as opposed to having people walking around going, well, you know, Susan over there is just not really holding up her own. I wonder what's wrong with her. Well, let's find out what's wrong and see if we can help Susan.
So most of us are Birch Scholars getting ready to take the next step into moving into the next step of our career. So what would be a wise piece of advice as we move into taking that next step?
Well, first of all, you want to find a program that's going to bring you on board, that is going to value your potential as an independent investigator, and going to protect your time so that you can pursue that and provide you with the resources that you need to be successful. And that would be things like space and some financial support and opportunity to build a team of your own, because it's going to be pretty hard for you to make progress if you're N equals 1 in most of the things that you probably want to do. So absolutely critical as you go around looking for where you want to land to ask those questions. And I'm particularly concerned about the one that oftentimes doesn't get fully laid out and then becomes an issue, which is how much of your time is protected to do research. If you're somebody who has clinical duties, are those clinical duties suddenly going to mushroom to become more than half of your time, and research gets squeezed over to the side because clinical duties sometimes are the way that the institution makes money. If you're not a clinician, but you're somebody who's going to be doing teaching, well, how much teaching are you expected to do? How many courses per semester? How many hours each week is that going to take to prepare and do the lectures and grade the responses and all that? I would say if you're going to be a successful researcher and compete with everybody else, whatever your field is, you need more than half of your time focused on research. If it drops below 50%, it's harder and harder to have your momentum sustained, especially right now when there's lots of other people whose momentum is going great guns, and they're your competitors as well as your colleagues. You don't want to be left in the dust by the whole field moving ahead while you were teaching or taking care of patients and your research program had kind of slipped into the background. Got to have that protected time. And while you may believe that, you got to be sure whoever's hiring you believes it too and is willing to commit to that for the first several years that you're there, that you are not going to be expected to serve on every committee and teach every course and take care of every patient and do all those other things that are going to distract you from your goal, which is to make a research career really get off the ground in the best way.
Awesome. Wonderful. Thank you. I know Dr. O has a question, so I'm going to turn it over to him to ask that.
Thank you, Erin. Thank you, Dr. Collins. As a faculty here at Emory, I spend a lot of time working with junior investigators. I know NIH has a lot of programs and plans to encourage and to support junior investigators. Given the funding level over the past several years, there's been a lot of depression in morale. I don't know if you can take this opportunity to let us know what NIH is doing to really stimulate the career of junior investigators, so that those that are coming behind.
Ingo, thank you for the question, or Dr. O, I guess, as we are now calling him. But to me, you'll always be Ingo. This is a great question and a great moment for me to answer it. We have, over the course of the last 15 years, seen NIH support for all stages of investigators get under some stress. You may remember we had this doubling of the NIH budget between 1998 and 2003, which was great. But then at that point, everything kind of went flat, and even worse than flat in some instances with something called the sequester. As a result of that, early stage investigator coming to NIH with their best ideas for the first time, trying to get their grant supported, we're encountering tougher and tougher odds working against them. This is not a good thing. We really want to be sure that of the entire part of our workforce, the part that I'm most concerned about is those early stage investigators. You all are our future, and we want to be sure that you're encountering a system that allows you to get started and then sustain a program. We did something 4 or 5 years ago that helped, which was basically to say, anybody who's coming to NIH with their first grant application, in the peer review process, they should compete against each other and not against the very experienced investigators who've written lots of grants and gotten lots of awards, who therefore are a little more likely to write a grant that's going to make peer reviewers think, yeah, we want to fund that. That helped in that it gave early stage investigators several points in the priority score effectively by having that competition done that way. But it's not good enough. So just in the last 2 weeks, we have announced a new program, which is not without controversy, but which I think you all will like, which is to say we've got to do one better than that. We've got to make sure that somebody coming to NIH with their first grant application has an even better chance of getting funded and not just missing the pay line by a few points. And we also need to think about those people who did get that first grant, but now they're back for their first competing renewal. Or maybe they're coming back with a new idea and coming back for that second grant. Those are our future as well. And those people have been getting squeezed also with the tight timelines. So we have decided, we corporately, all of the institute directors and myself, that we're going to redistribute funds from other places and come up with $210 million every year starting this year. This is already happening for the foreseeable, to allow those applications that we're going to miss the pay line to get funded. So that's about 200 new investigators and about 200 of these who are what I would call early established investigators who are ready to kind of take off but might not be able to if they just miss their pay line on their competing renewal. That is a pretty big step. The people who are not happy about it are the people thinking about, wait a minute, where's that money going to come from? And especially investigators who maybe have already a lot of grant awards and are happy to keep on getting more. We can look at their productivity. This has been an interesting bit of data collection. And when a lab gets to a certain size, adding additional grants and additional people, dollar for dollar, the productivity doesn't quite stay as strong as it was when you were at a modest size. So part of this is to try to look at those data with an unflinching eye and say, maybe that's not the ideal way for every NIH dollar to be spent. We should take some of those money and give that to the next generation. So this is called the Next Generation Researchers Initiative. If you want to go to NIH website and look that up, you will see exactly what we are doing. And it is already having consequences this month with a whole bunch of investigators who thought they weren't going to get funded, and now we can just barely figure out a way to make it happen. About 400 grants of this type altogether are being funded this month at the end of fiscal year 17 that otherwise would have gone unpaid. Cool, huh?
That's awesome. I know that's music to all of our ears. Thank you for sharing that. That's very exciting. I didn't realize I was supposed to pull up this separate menu to get the question, so I'm going to pull a question from one of our participants. I don't know who it is, but they're a junior faculty that applied for a K award that involved a team approach and learning different techniques to answer the proposed hypothesis. The biggest critique of his or her application was that the project relied on expertise of too many people. How do you suggest the junior faculty members start to distinguish themselves from the mentoring team?
That's an interesting one, and I'm not sure quite how to react to it without knowing more about the details, because we've just been talking about how wonderful it can be and how productive it can be for a junior faculty member to take part in a team. Clearly, in this instance, the reviewers thought it went overboard and there were too many components to this. Maybe it didn't have the focus they were looking for. So again, in terms of distinguishing yourself as the junior faculty, that's a discussion you got to have right up front and make sure that the mentoring team is completely on board with that as a critical part of the team coming together at all. And if that wasn't clear in this instance, and it sounds like it wasn't to the peer reviewers, maybe that was a step that, if it happened, needed to be better documented.
And then I know we're starting to wrap up here a little bit, but most of us are women's health researchers, being that we're Birch Scholars. So what do you think are currently the 3 most important research priorities in women's health right now?
It'd be hard to name just 3. Again, I'm really happy that here at NIH we have the Office of Research on Women's Health, and Janine Clayton, who's the current director, does a terrific job putting in front of all of the other institutes opportunities in that space. I think these range from basic science to clinical. I will say, when it comes to basic science, the focus that we have now managed to insist upon, that you look for sex differences when you're doing an animal model experiment, which all too often was being neglected by people who only studied male rats or male mice. We're not allowing that anymore. We've made a big change there. And we're learning interesting things about sex differences that maybe very well could apply to humans as well, just by making sure we did the animal studies right. When it comes to the clinical studies, I think we now have a requirement, NIH does, that any phase 3 trial has to not only enroll women and underrepresented minorities, but has to be designed to do the analysis to look for differences between those groups, not just collect the data, but actually ask the question, is there a difference? And if so, to try to sort out why that is. Those are really important areas and areas of opportunity. I think sex as a biological variable has oftentimes, in many different quarters of what we do, even cell culture experiments where people don't know what was the sex of that cell line they were studying. It's been ignored. And we have the opportunity now, with increasing consciousness in this space, to apply that across a wide range of experiments. So I would say in women's health, we have the chance to do this. As far as specific clinical applications, I continue to think about heart disease as an area where we still, though we enroll a lot of women in clinical trials on heart disease, it doesn't seem like women's heart disease is quite the same as men. And we need to be really thoughtful about the design of the trial and whether we're capturing those differences simply by who we invite to take part. That is something I know the director of the Heart, Lung, and Blood Institute, Gary Gibbons, is quite interested in attending to. And that kind of thing, very much, I hope all of you, you Birch folks, will also be contributing to, because you're our eyes and our ears and our brains when it comes to where we need to go in women's health. That's what this program has been able to do, is to create this wonderful critical mass of people who are interested in these issues and know how to do the right research to answer questions.
Thank you, Dr. Collins. And I'm just going to, I know we're running out of time, but I want to ask one last question from one of our participants. And as the director of the NIH and being such a public person, can you talk about your strategies for best communicating science to the community and how you train your team to do so?
Yeah. Well, first of all, it's up to all of us. The idea that the researcher can go to the laboratory, close the door, and count on somebody else to be the communicator, that model ain't going to work. It never really has, and it certainly won't now. Yeah, I would think all of us as scientists need to be prepared in a sort of elevator speech model, if called upon, to be able to talk about what it is that we're working on and why it matters, so that a random person on the street or your grandmother would understand why this is so exciting, why it's so important, why you're putting your life into it. And then if you make a discovery of some sort that becomes of interest, even to somebody in the press, you're prepared. Not to overstate what it means, but not to confuse everybody by using a lot of jargon either. Being able to articulate the nature of the science you're doing and what it means for our understanding of life and disease and the nature of what it is to be human. All of those things, I think, need greater emphasis. I think we're doing a little better job now in our training programs to give people that opportunity to learn those communication skills. If you don't think you've had that opportunity, look around you in the institution where you are, find the people who are good at that in the communications offices or the training offices, and get yourself trained. Many of them will give you a chance to do a mock interview with a press person with a video camera running, and then you can look at it and say, oh boy, I probably didn't do that quite the way I meant to. I can work on this. Work on it. Be prepared. Don't get caught off guard.
Oh, thank you, Dr. Collins. I know I didn't get to all the questions, and I apologize to the participants for that, but we are so honored to have had this hour with you and are very grateful. So thank you so much for sharing your expertise with us.
It's been a pleasure and a privilege. And again, my hat's off to all the Birch folks for the great work that you do. It's nice to be able to talk to you for this brief hour.